My name is Long, and I'm wrapping up my degrees in immunology and computer science at the Johns Hopkins University. So today I'll be introducing you an algorithm we recently developed called Tracer. So it is for a technology called spatial transcriptomics. And frankly speaking, spatial transcriptomics is transforming how we map the tissue biology, but almost every downstream analysis assumes that the transcript-to-cell assignment is already correct. And my work starts from the opposite premise in real tissues, especially the thick, dense volumetric tissue. The cellular profile itself is often the first source of errors. So Tracer is designed to correct that error directly from the spatial transcriptomic data itself. So this is a whole transcriptome ADARA cervical cancer data set with hundreds of millions of transcripts. So when we compare an orthogonal problem score from this recent tool called OverPy, which measures the vertical structural integrity with tracer conflict score. So both highlight structured regions of discordance. These are not random noisy spots. They follow like a very delineated tissue architecture. So the key point here is that transcriptomic discordance is spatially organized, and that means it is not just a downstream annotation problem, it is actually a cellular reconstruction problem. So here in the instance of the Z-stack series, you can see that this region, which has a dominant gene expression program of the T and NK cells characterized by the CD3 family gene expression, is also concurrently expressing genes that are only restricted to the myeloid lineage, such as the canonical marker genes of CD68. So both OverPy and Tracer measures different things, and the fact that they have spatial agreement here tells us this problem is real. And this issue is really not specific to one technology. In sequencing-based spatial transcriptomics, such as VZM-HD, the starting objects are bins rather than segmented cells, and rigid binning can also mix neighboring cell states or split one biological cell across multiple bins. And here, RCTD-based ambiguity and tracer conflict again reveal spatially organized uncertainty. And for the H&E insights, you can see that in this region, where the dominant gene expression program is the mesangial, it also concurrently expresses conflicting marker genes that are only restricted to distal convoluted tubule cells and thick ascending limb cells. So the broader problem here is that spatial transcriptomics gives us transcript coordinates or bins, but the biology actually lives at the other level, the level of cellular profiles. So here is a conceptual failure mode. A tissue section is not a perfect monolayer. So it is a thin slice through a 3D cellular volume. So some cells are cut through the nucleus and are very easy to segment. Some cells are only partially captured without a nucleus. These are what we call like anuclear cells or partial cells, and other cells overlap in X and Y due to the 2D projection, but are perfectly separable in Z-axis or by transcript coherence. So most existing segmentation approaches borrowed from computer vision field are forced to decide, assign transcripts to the nearest nucleus, leave them unassigned, or bin them. Tracer asks a different question. Do the transcripts assigned to an object form a biologic coherent entity? So Tracer has two operating modes. In the segmentation mode, it starts from an existing segmentation mask and refines it. In no seg mode, it starts from bins and reconstructs cellular profiles without a prior mask. So the core idea here is simple. The transcripts from the same biological cells should be spatially proximal and transcriptionally coherent. Tracer therefore learns a coherence prior from the data, prunes the conflicting assignment from cells, and rescues the residual coherent residuals and group them into partial cells and stitch profiles when there is a strong evidence that they belong together. Importantly, tracer is reference optional, meaning that you do not necessarily require a single cell reference, cell type labels, or marker genes to run. And it's also worth mentioning that low quality or incoherent transcripts tend to be demoted by the coherence model. So tracer reduces the reliance on the aggressive manual filtering of low quality transcripts. So this is a PDAC, a pancreatic cancer Xenium immuno-oncology IO panel with over 20 million transcripts. The full tracer test runs on orders of minutes on a laptop without HPC or GPU. So this matters because the spatial analysts often spend more time fine-tuning the hyperparameters than running the algorithm itself. And the alluvial plot on the right here is useful because it really just makes tracer auditable. We can see transcripts that remain in the original cells being reassigned to the neighboring overlapping cells. Transcripts demoted as incoherent, and transcripts reconstructed into the new partial cell profiles. And this is not simply filtering, it is a redistribution of transcript evidence into more coherent cellular profiles. And here I'm going to focus on two regions of interest, two ROIs. And the first ROI on the left, both refined whole cells and reconstructed partial cells form structured transcript clouds rather than diffuse noise. That's the first sanity check. These are spatially coherent entities. And on the right, after tracer, we recover immune partial cells covered by the white mesh grids that were not represented as separate cellular entities in the original 10x segmentation. So this basically creates new immune tumor proximities that would be missed if we only analyze the original 10x segmented cells. So the key biological implication is that segmentation errors do not just change the cell counts, they can erase the cell-cell context that we use to infer, for example, the tumor immune organization, which is the whole initiative why we choose to use any spatial technologies. And in sequencing-based spatial transcriptomics data, such as Visium HD, there's like no true cell boundaries. And the data starts from, usually 2x2 micrometer bins or 8x8 micrometer bins. And existing strategies either keep bins or aggregate them, but the rigid binning do not necessarily correspond to any cells. So in the no-seg mode, tracer starts from one single pixel and reconstructs the coherent cellular profiles using the same spatial coherence principle. So this makes the method platform flexible. With a segmentation, tracer refines and reconstructs. Without a segmentation, tracer builds the profile from the ground up. So this is a normal kidney where we have strong anatomic structures or expectations. The glomeruli here, tubules here, and stromal compartments. So tracer reconstruction preserves these known anatomic structures. And the important part is that it also recovers the overlapping or partial cellular states that are hard to represent using the rigid binding method. So in this ROI example, tracer separates the cell type specific profiles that are spatially interleaved at the band level. So the no-seg mode is not just making prettier maps. It reconstructs the profiles that align with the tissue anatomy and the marker structure. So after showing the qualitative anatomy, we benchmark tracer against the 10x based ranger segmentation and the bin-to-cell. And I'm not gonna walk you through every single metric here, but like the key takeaway here is that tracer at 2 micrometers shows stronger marker signal structure and improved RCTD style purity and a higher correlation with the single cell reference. And the important point is that not one metric win everywhere. The important point is that tracer improves cellular coherence while preserving biological expected cell type programs. And for sequencing-based platforms, this suggests tracer can bridge the gap between the pixel scale measurement and the cell scale interpretation. So we next benchmark the segmentation mode using a non-small cell lung cancer small xenium sample against several recent approaches. And it's worth mentioning that these methods are not all designed for the same purpose. And for example, Baser, ProSeg, and Sager are transcript segmentation methods, either relies on probabilistic models or deep learning, graph neural network. And CellAdmix and Split address different forms of cell profile cleaning. And tracer focuses on the refinement and reconstruction. And in this setting, tracer is the fastest and the most memory efficient while achieving the strongest RCTD-based purity metrics and competitive marker specificity enhancement. So practically speaking, this means that tracer can be inserted after existing segmentation rather than replacing the entire upstream pipeline. And then to test the generality, we extended the benchmark across ROIs from ELRA, Xenium 5K, Cosmix, and Merfish. So these platforms differ in chemistry, transcript density, panel size, and starting object definition. So across platforms, Tracer's most consistent advantage is the runtime and the peak memory and the marker specificity enhancement. And it's worth mentioning that Split performs very well on reference-based concordance metrics, which is expected because its objective is closely related to RCTD-based deconvolution against the reference single-cell RNA-seq data. So here, the broader message is that tracer is not a platform-specific patch. It is a general coherence-based layer that can sit on top of multiple spatial technologies. So once the cellular profiles are more coherent, the next question is whether that changes the biological interpretation. So here I'm focusing on the VISTA VSIG immunoregulatory axis because VSIG family ligands, including VSIG4, are macrophage-associated checkpoints that may shape the T cell dysfunction in the solid tumor microenvironment. So this will give us a concrete test case. Can reconstruction reveal immunosuppressive niches that were underestimated by the original segmentation. So back to the PDAC xenium IO sample, tracer improves the marker specificity and identifies a large population of confident partial cells. Among these, macrophages and fibroblast associated partial profiles are prominent, consistent with the dense stromal and myeloid structure that are commonly observed in the PDAC samples. And focusing on the VSIG4 positive tumor-associated macrophages, TAMs, tracer increases the fraction of VSIG4 attributed profiles and brings these macrophages closer into closer proximities with T cells compared with the original segmentation. So this suggests that inaccurate cell profile reconstruction can obscure the immunosuppressive niches, and we are currently validating this axis in related PDAC Visium HD samples. So, so I'm just gonna quickly go through this because this is originally developed for the breakthrough cancer GBM data, team lab data. So there are some ongoing work with like, incorporating part of these GBM biology into the manuscript. And and with that, I would like to just like summarize the talk. If there are only two takeaways that you want to remember from this talk, I would like to say the first thing is really just like my name. Remember that. And the second, the one sentence take-home message is that better cellular profiles enable better spatial biology. And the central message here is really that spatial biology depends on the cellular unit we reconstruct, and tracer provides a reference optional way to make that unit more coherent, more complete, and more biologically interpretable. And with that, I would like to thank all my collaborators across all these institutes within the Breakthrough Cancer Consortium. And Tracer is actively being deployed on the multiple team labs data set. So I guess stay tuned and hopefully you will see Tracer a lot in the future.